for peace, light a candle for love, light a candle that shines all the way around the world. Light a candle for me, light a candle for you, that our wish for world peace will one day come true. Thank you. Without further ado, let us introduce ourselves. I am Vaishnavi Guru from class 9.
I'm Ananya Dixit from class 8.
We are the main curators of this symposium.
Next, we are Sejal here to present the welcome note.
Distinguished guests, participants and viewers, a warm welcome to the Virtual Science Symposium hosted by Vidyadhiraja High School and organized by the Global Young Researchers Academy GYRA. This symposium is a platform for all of us to learn, inquire and share our passion for research. I extend my heartfelt gratitude to GYRA for giving Vidyadhiraja High School an opportunity to host its first symposium. We are privileged to have Dr. Finosh Thankam, founding president GYRA, as the chair of our symposium. Dr. Finosh Thankam is an assistant professor in tissue engineering and regenerative medicine at the Department of Translational Research, Western University of Health Sciences, California, USA. On behalf of Vidyadhiraja High School, we are privileged to extend our warm welcome to Dr. Finosh. We are delighted to have a distinguished guest speaker, Dr. Irimpan Mathews. Dr. Mathews, co-founder GYRA, is a staff and lead scientist at the Stanford Synchrotron Radiation Light Source, Stanford University. On behalf of GYRA and Vidyadhiraja High School, we welcome you. Your insights will enrich our symposium, Dr. Mathews. I would also like to welcome Shri K.N.S. Nair, the founding president of Vidyadhiraja High School and Junior College, whose steadfast support serves as the cornerstone of our institution's success. A warmest welcome to Team GYRA for their immense commitment and unwavering vision in creating next-generation scientists. A hearty welcome to all participants, our coaches Ms. Helen Roach, Mrs. Babita Anand and Mr. Sharad Palkar, audience, students from other schools and teachers who are viewing this symposium on YouTube. As we begin the session, let us remember that every idea shared and every question asked contributes to the collective pursuit of knowledge. Thank you, Sejal. Now, with great honor, I request Shri K.N.S. Nair, President, Vidyadhiraja High School and Junior College, to extend heartfelt felicitation to GYRA.
Distinguished speaker, Dr. Irimpan Mathews, Dr. Fino, principals and other members of the GYRA family. It's with great pleasure that I'm here before you today at this symposium. We are gathering up brilliant minds, curious learners and passionate students. The collaboration between Dr. Fino and the VHS family is a partnership that opened up in 2019 and has flourished since then. This enduring commitment between our institutions has also paved way for groundbreaking projects by our young learners, such as the Water Security Project, which reached the state level. Dr. Fino's guidance and dedication to training students at VHS have instilled within them a scientific attitude. It has empowered our children to think with innovation and precision. The participation of VHS in prestigious events like the Science Congress demonstrates how we are expanding our horizons beyond traditional academic realms. I extend my heartfelt gratitude to GYRA for offering VHS the opportunity to host this debate symposium. I must also mention here the commitment and enthusiasm on the part of our principal, Mrs. Lakshmi Sojit, in promoting the activities of GYRA in VHS and the schools around. As we gather here today, united by a shared passion for scientific inquiry and progress, let us also look forward to the future with anticipation and enthusiasm. With immense faith in the capabilities of our young researchers, I am confident that VHS will continue to thrive and excel. We eagerly await the prospect of hosting IRCC 2025 in Mumbai. Together, Let us embark on this journey of scientific knowledge, research, and collaboration as we move to even greater heights of excellence. Thank you very much.
To guide us through the symposium, we have the distinguished Dr. Finosh Thankam, founding president GYRA, as the chair of the event. Master Samya will share a brief introduction of Dr. Finosh.
Ladies and gentlemen, esteemed guests, it is my honor to introduce Dr. Finosh Thankam, our esteemed chair for this event. Dr. Finosh is an assistant professor in tissue engineering and regenerative medicine at the Department of Translational Research, Western University of Health Sciences, Pomona, California. He is the founding president of GYRA. Throughout his eminent career, Dr. Finosh has demonstrated an unwavering dedication to advancing biomedical science, particularly in the fields of orthopedics, cardiovascular diseases, and tissue engineering. He is deeply committed to utilizing his expertise to benefit millions of individuals afflicted with various diseases, especially those facing challenges with musculoskeletal disorders and cardiovascular diseases. Dr. Finosh's academic journey is marked by outstanding achievement. His research contributions span across numerous areas, including molecular and interventional cardiology, stem cell biology, biomaterial science, and proteomics. As an accomplished scientist, Dr. Finosh has contributed significantly to scientific studies with more than 100 publications in esteemed journals. His passion for teaching and mentoring is evident through his dedication to nurturing the next generation of researchers. It is a privilege to have him as the chair of our symposium. We look forward to his invaluable insights and contributions. Please join me in welcoming Dr. Finosh to address the audience.
Thank you, Soumik, again for this wonderful introduction. I would like to apologize everyone for taking your time due to little bit technical glitch occurred. So kindly pardon us. Yeah, we are back. My goal today here is to mention this symposium. We established an organization called Global Young Researchers Academy, GYRA. Headquarters is in Portland, United States. Our mission is to foster scientific temper and research aptitude in school kids, especially in middle school kids. Our goal is to train the school kids with proper scientific research methodology so that they can avail world-class research avenues in the future. And we keep no barriers to the research. The students can find any projects of their interest, and our team GYRA will be training them to identify a solution, identify a problem, and to find a solution for those problems. Our dream is very simple. Our dream is to double the pool of global researchers in the next 50 years. So we work as a team. Our team includes scientists, engineers, educational professionals, and people from different sectors of life. Before the concept of GYRA was conceived, we as a team have done extensive homework as well as several pilot projects in several schools as well as neighborhood communities in India and United States. Vidyadhiraja High School was one of the first generation schools to accept our GYRA mission and gave us a wonderful opportunity to begin GYRA projects in their premises. As Nair sir mentioned before, we started working with Vidyadhiraja kids since 2019, and we were able to generate immense scientific curiosity in their schools. So now we are trying to expand these lessons to other schools, if possible, across the globe within the next 20 to 30 years. So far, we have more than 1,000 kids engaged in various research activities in India, Singapore, Middle East, United States, and other parts of the world. On behalf of my team GYRA, I extend my sincere gratitude to Vidyadhiraja High School to giving us an opportunity to launch our first virtual symposium. And also my sincere thanks to the vibrant students and teachers of Vidyadhiraja High School. Today's symposium consists of two parts. The first part will be a talk, a lecture by an eminent scientist, which will be followed by the presentations by the GYRA scholars of Vidyadhiraja High School. We will be having 3 presentations from the school kids. Once again, thank you all for rejoining for this symposium. Kindly accept our sincere apologies for the technical glitch. Please take a moment to subscribe our YouTube channel if you didn't do so. And again, this is a virtual platform. We may encounter such network glitches, but we will try to resolve and come back as quickly as possible. So once again, welcome to this symposium. Thank you again, and back to MC.
Thank you, sir. A famous quote goes like this: "Science knows no country, because knowledge belongs to humanity." It's time to introduce our esteemed guest speaker, Dr. Irimpan I. Mathews. Liana Leo, please.
Good morning, all. It is my great pleasure to introduce to you Dr. Irimpan I. Mathews. Dr. Mathews is a distinguished scientist whose career has spanned continents and various disciplines. He is a lead and staff scientist at Stanford Synchrotron Radiation Light Source, Stanford University. He has left an indelible mark on the field of structural biology. With a PhD in crystallography from the Indian Institute of Science, Dr. Mathews has a treasure trove of experience gained from prestigious institutions such as Michigan State University, Cornell University, and Stanford University. His contribution to science, especially structural biology, are vast and impactful. He has made magnanimous contribution on the fields of protein crystallization, enzyme mechanisms, and structural studies of critical human pathogens. His work as a research associate at Michigan State University under the direction of Professor Tulitsky led to the publication of important papers describing proteins involved in blood coagulation. Notably, his research on the flavin-dependent thymidylate synthase has shed light on DNA building blocks crucial for the survival of organisms. Furthermore, Dr. Mathews has been at the forefront of intense scientific collaboration, working with esteemed colleagues on projects ranging from polypeptide synthase to studies. Research has resulted in groundbreaking discoveries with far-reaching implications. He has published more than 100 scientific articles in his field. Beyond his scientific achievements, Dr. Mathews has also played a crucial role in addressing global challenges. Notably, as a member of the National Virtual Biotechnology Laboratory during the COVID-19 pandemic. In addition to his research endeavors, Dr. Mathews is a visionary in the design and fabrication of novel devices for crystallization and synchrotron beamlines, contributing to advancements in experimental techniques and data collection methods. Dr. Irimpan Mathews exemplifies excellence in scientific inquiry, collaboration, and innovation. It is with great admiration and anticipation that we welcome him today. And I have no doubt that his continued contributions will shape the future of structural biology and beyond. Please join me in extending a warm welcome to Dr. Mathews. In the words of Marie Curie, nothing in life is to be feared. It is only to be understood. We eagerly await Dr. Mathews' insights during his guest talk. I would like to call upon the chair to invite Dr. Irimpan Mathews.
Thank you, Ananya, for your nice words. This is the time to begin our virtual symposium. I am very privileged and humbled to welcome Dr. Irimpan Mathews from the world's #1 university, the Stanford University. Dr. Mathews has been a strong pillar for GYRA since the conceptual level, and he is currently the Vice President of GYRA Global. And Dr. Mathews has done outstanding research contributions, as you mentioned from the, as you listened from the introduction. And one of his recent findings received FDA approval for cancer therapeutics. I'm sure those findings will be going to help millions of sufferers, or bringing back millions of cancer sufferers back to their life. I would request the audience from the YouTube to post your questions in the chat. Our volunteers will be sharing it with Dr. Mathews towards the end of the presentation, and he will be discussing his insights and thoughts to answer your question. So there will be a Q&A session after completion of his talk. And today, Dr. Mathews will be discussing about our immune system, the fundamentals of immune system, and how those understanding has led to certain findings in the medical field. His talk, entitled X-ray View of Immunology, will be taken place now, and it will be a 30-minute talk followed by a Q&A session. With my great pleasure and honor, I welcome Dr. Mathews for the talk. And Dr. Mathews, the floor is yours. Thank you. And thank you again for your patience. Bye.
And thank you, Pinosh. Thank you, Mr. Nair. And also very special thanks to Principal Lakshmi teacher. And also let's thank Liana for the introduction. Let me share my screen. Can you see the screen?
Yes, it is visible.
No, just no, we were running for time, and we were seeing the issues of the time. One thing, we have a lot of research students here. The research is the only one that is not driven by time. It is driven by passion. Passion drives research. When passion drives, pain goes away, and you will find time. Yes. Today's talk is about X-ray view of immunity. My name is Irimpan Mathews, as Liana said, and this is a GYRA Symposium, and I represent GYRA and SLAC National Laboratory, it's a national facility, and Stanford University. First, we'll see what is X-ray view. You know, you probably have went through X-rays a few times if you have went to dentist or when flying to the X-rays. Now I'm going to take a simple example of what is X-ray view. Have you seen this? This is a radio, isn't it? Yeah, and people use it for different things. Here you can see one person is listening to music, other one probably listening to a news. And here I have Jim. He is not really listening to something. He's trying to think about something. Actually, he's thinking about what is inside this radio. And what he does, you know, remember your parents are there. Therefore, he hide under a desk and try to look the radio. And he opens it up and he comes to different parts of this radio. Now I am going to introduce you another radio. This is called immunity radio. It looks a bit fancy, isn't it? Very interesting. We'll try to look and see what are things inside. And then open the cover and you don't see many things. And suddenly you see a clock thing. This is a clock kind of radio. And let's look at the numbers. Instead of numbers, there are objects. There are various objects around the radio. And we want to see what is the inside one. See, there's a diagram there. There are 5 different things. And if you look one of them, it is this complex. It's very, very complex. And each of these things are like big enzyme. Therefore, we are not going to go there. Actually, you can see immunity is a very complex thing, but we are trying to see very simple picture of immunity. Let's see what are these guys. The T cell is the lymphocytes, phagocytes, neutrophil, eosinophil, basophil, mast cells, and platelets. These are the different players in this field. Now, have you seen this? This is a bone marrow. It doesn't look very nice, isn't it? It looks, oh, that looks awful. But here you see, this is a really good food made from bone marrow. Why am I talking about bone marrow? See, all these guys are generated in the bone marrow. You can see this small box of each of these units. Therefore, they are all made in bone marrow. Now, let us look and see what are these things called. All these things collectively called white blood cells. And except these platelets, they are cell fragments. Now, try to see what they do for us. They protect body from infection, help cancer, the T cells and all guys. Then you can see here phagocytes and things like that. They surround and kill microbes, ingest foreign materials and remove the dead cells. We're going to take an example here. Here you can see this is a small cell. This is the monster. This is the one we want to kill it. This is from an outside source. For what you'll do, it will try to take it inside, then try to cut it. It's a specific enzyme called the lysozyme. They use lysozyme and cut it into pieces and take it out. The process is called phagocytosis. I'm going to show you a live example of this. So hope you can see the picture. Here what is happening is cells are trying to, the guy on the top, that was the bacteria from MRSA. This is a methicillin-resistant bacteria. That's how it's coming to engulf the bacteria and then kill it off. Now, this small thing inside this, this thing is called phagosome. And neutrophil, they are the ones very rapid response team, and it's most abundant in the cells, in our blood. And other side, this mast cells and these guys, they are affecting or regulating allergic responses and the innate and adaptive immunity. We are going to see what are those things. And platelets, as you probably know, they are the ones stopping the bleeding. Now let us look at different ways of immunity. One is called innate, inborn, native, or what you mean. These things are there with you when you are born. And other one is acquired or adaptive or learned immunity. You learn it probably sometimes from vaccines or maybe from bacteria and things like that. And as you can see, this will be the first line of defense because they are in your system. And this will be the second line because they have some learning process to get there. But these are the immediate and maximum response. And there is some delay because these guys have to learn. And where do you see these things? Epithelial surfaces, respiratory tract, those things are very, very important because when something is, some foreign material is getting, it can take all these guys and take it to, send it to the stomach. There it will be killed. And the tears, they have the, tears has lysozyme in it. And that is going to clear a lot of bacteria. And here, through infection of microbe, sometimes you may say it's not good, isn't it? But sometimes you learn things from the microbe and you get better activity against them. And when you think about the immune system, you want to have a very perfect system, isn't it? Because if you have a very perfect system, you don't have to worry. Your life is very good. Therefore, we want immune system to be very perfect. For that, what we need, the basic thing is, see, immune system, that's the really killer kind of thing. Therefore, immune system has to know what is yours and what is not yours. See? Therefore, what they have is, in our body, we have a school. A team from Harvard, they found something very interesting. There is a thymus gland that teaches and trains cells. It's like a small elementary school. Like you see, they are going to teach everybody whatever is needed. What are they going to teach? They educate new immune cells by exposing them to proteins made by thymus cells that mimic various tissues throughout the body. What's the result? Whatever is coming out from this thymus time as a school will know everything about your body. For when a foreign material comes, the system will know they are foreign or is yours. Okay, this is very, very important. Now, let's summarize what we learned so far. We talked about the radio. Remember, Jim? He was trying to go inside, and that's what we did. We took this in immunity radio, and we went inside and trying to find out what are those guys. And we talked about immunity types. Some of them are inborn, some of them are we learn from outside. And then we talked about the school, the time is a school where students were, each and different cells were learning our system and different system. And now here, see, usually when you go outside, we don't care much, isn't it? We are enjoying the life. But if you look a compressed piece of air, say air compressed, you can see a lot of things inside. You won't believe me, but in the COVID time, now you know, because you are using a mask because air had some things inside. We want to avoid them. Therefore, air is not pure. And look at the water. If you really look in the water, there are various things inside. And generally, we don't worry about these things. You know why? Because we have the immunity against all these guys. That's why we don't worry. For it will be interesting to see what is the mechanism, immunity system. Let's see. Here we were talking about these things so far. Now we are going to go a little bit deeper and trying to see what are inside these things. Okay. I have to learn you. I'm going to introduce 3 different things that I want the students to learn. Okay. One is antigen, one is antibody, and other one is cytokine. Okay. Let's see what are these guys. Antigen is a foreign substance. That means it is not from your body. And antibody are the protective proteins. And cytokines, they are signaling proteins. They are very, very important. See, they tell immune cells what to do to keep immune system function correctly. It's very, very important. Now, let us look at a little bit more on the antigen and antibody. Antigens trigger immune response. Therefore, these foreign guys, when it comes to system, the immune system starts operating. And antibodies are lock on to specific antigens. Antibodies, what they do, they neutralize it, and they also activate other proteins. If they are not able to do things, then they will introduce other guys to really work on it. Now, antigen, they are the ones substances can produce any immune response. And there are various antigens. Some of them are outside, some of them are inside, some of them are tumor antigens. There are various antigens. Or they're coming from pollen, bacteria, viruses. All those things are antigens. And the antibody has to be very specific for each antigen. Or it will be affecting all those guys, isn't it? They have to be very specific. Therefore, we are talking about interaction at molecular level. If you look at my hand, if my hand has to do something, various different things, it will be better for the fingers to do it, isn't it? Or if it whole hand does it, sort of energy is involved. Therefore, most of the time, the interactions happening at the molecular level, we'll try to see what are those interactions. One very interesting thing is something called antigen presentation. See, when the antigen is coming, it's getting presented by somebody. Let's look at some of the presentation. Here, somebody is presenting a horse, okay? And here, another person is presenting a horse. What's the difference? The first guy, let me put a pointer, this option. The first guy, he's doing it in a very stylish way. She's doing it in a very simple way. Now, for the body, we have antigen presenting cells. And I'm going to give a cartoon diagram of antigen presenting cells. These are antigen presenting cells. And where do they present the antigen? They want to present to the yama of this thing. He wants to kill these guys, isn't it? That's where you want to present it. And they can't present it everywhere. They have specific places to present, either here, here, or here. Therefore, you have to be very specific. And for that, this antigen presenting cells will come. Here is okay. See, he can't really reach there, isn't it? Then they will try to put something extra to reach. Now he can reach, or he can make it more fancy, add more things, and then reach it very well. Then reactions will be much better. If you're curious, These things are called, this is called major histocompatibility complex. It's a big protein. And this is the T cell receptors. These are the places reaction is happening. And these are the various proteins involved in making the reaction go faster. Now, in the importance of this is initiates appropriate immune response because of this reaction. For what we'll do, all these bacteria will try to attack this. They try to disable the system so that they can survive. Now let's come back to this antigen. And see, when you look at the antibodies, they have to lock into each of these guys, isn't it? Now let's just take some example here. You know, when you go outside, sometimes some seeds or something's attached to your body, isn't it? They attach in different ways. And here for the antigen, antibody needs to lock onto several different types of antigens. Remember, when your mom will say sometimes, no, if you put this cloth, they may get more seeds on it. Something will attach. They have to put better different kind of clothes. Similar kind of principle here. When the antibody needs to lock to different types of antigens, they have different mechanisms and a very specific locking mechanism for each antigen. Now let's take a thinking process. What are the benefits of studying the details of the antibody interactions? See, we are talking about, for example, if the bacteria comes in and we are trying to look at another system in the body which can deactivate the bacteria. What's the advantage of studying these things? Or think about the cancer cells. Your cancer cells coming and you are trying to have system to block it. What will be the advantages? Let me make it a little more clear. See, if you have a gloves made of wool, if that attaches a lot of these seeds, you can make a gloves made of satin or silk or something like that. Then no, or some things which will not attach these things. Similar things can be happening here. See, if you really learn this process, you can use it for cancer, heart diseases, or multiple sclerosis, organ transplant. So organ transplant is a really big issue because you are trying to put a different organ into your body. Then immune system knows they are not from your body, isn't it? Then they won't accept it. And you have to learn the process very well to make them accept. Now, talk about antibodies. They are immune produce antibodies to fight off antigens. And antibodies are protein molecules. Now, if you don't know much about proteins, if you look at the last International Research Children's Conference, there's a talk about the protein molecules. And antibodies are Y-shaped molecules. I show you the shape like this. See, you can see this is a Y-shaped molecule. And antibodies have specific binding sites that binds to specific antigen. I got to introduce two terms. You don't have to really worry about it, but no, I'll get an idea of this. One is called paratope, other one is called epitope. We'll take an example here. Here you can see this is the antibody which is going to lock onto antigen, and here is the antigen. Therefore, this is the paratope and this is the epitope on this side. And here you can see this antibody is specific for this antigen. Now, if another antigen comes, it's not going to bind here. You see? And here, what are the antibodies do? They eliminate it, but sometimes neutralize it, and try to destroy it. If they can't do it, they will call other people to help. That is the antibodies doing. Now, little more details about antibodies, because they are very important, isn't it? They are the one protecting us. They are immunoglobulins, and they have two chains. One is light chain, other one heavy chain. You can see heavy chain and this light chain. And they're linked, basically means they're linked means they can't separate it out because they separate, then they don't function. You see, they're connected together. And both chains are divided into variable and constant region. We are coming to in a minute. And they have amino terminal as the variable region and C terminal is the constant region. I'll put an example here. Therefore, this is the antibody. Where is the amino terminal is on the top and the C terminal is on the bottom. And these are the regions which are variable, okay, near the amino terminal. Now, there are 5 different type of immunoglobulins. One is called the gamma, alpha, mu, epsilon, and delta. If you want to remember better, they're called gamed. Okay, done, gamed. Then you can remember these names. Let's see how they look. Gamma is the Y-shaped molecule, and it has 2 binding positions for the antigens. And the alpha it's a dimer kind of thing. It has 4 positions. And mu has it's a pentamer. It has 10 positions for binding to the antigen. And epsilon has 2, and other one also has 2 positions. What do they do? The gamma one binds to phagocytes, and it's the most abundant one. The alpha one is secreted into mucosa and saliva and tears. And the mu is the first one coming to scene to attack. And epsilon is responsible for allergic reactions. Similarly, delta is a signaling antibody. These are the one tells the other system to start on. Now, let us summarize what we learned so far. We talked about antigen, cytokine. What's the other one? Anybody out there? What is the third one? Did I hear somebody saying antibody? Yeah, that is the antibody. We'll check from the YouTube and see who are answered correctly. And we talked about the antigens, how various antigens, there are different antigens in the system, and said the antibody has to be specific to each antigen. And we talked about the antigen presentation, where systems are presenting the antigen to where, and these are antigen-presenting cells, and they are presenting to YAMA, the killer of the system. And we talked about these interactions, the importance of learning, understanding the interaction between the antigen and antibody to make better drugs against pathogens or maybe for cancer, metastasis, things like that. And we said these are the important areas where these things are applied now. And we talked about antibody, and it's a Y-shaped molecule, isn't it? And we talked about the antibody. Here is called, what's called? Paratope or epitope? This one is called paratope. Yeah, thank you. And we talked about different types of antibodies. Okay, there are 5 of them, remember, gamed. Now we are going to take one example. Okay, here this is a influenza pandemic. Now we are very familiar with the pandemic. After COVID, this name is very common. And here, this was a really big killer. It happened in 1918-20, infected 500 million people and killed 50 million. Some estimates say it killed 100 million people. And this is the virus. This is one term I want you to learn. This is antigenic drift. What does that mean? That means virus changes the antigen. Therefore, once if the antigen is looking like this, next time it looks like this, then what will our body do? See, body made the first antibody to disable the first antigen. Now virus brings another antigen. Therefore, our body needs to do a new level of protection. They have to make a different antibody. Therefore, what we do, every year we make new vaccines because we are expecting different kinds of virus. And here I'm showing the real virus antigen. Okay, and if you rotate it, it's a trimer. You got 3 sides, and this is called hemagglutinin, HA1. And this is a view from the top. Okay, and this is the changing variable region. I'm putting it here, and from the top, this looks like this. Then what we do, we usually make the medicine at the variable region. This is a variable region, and you can see we made this medicine. These are the vaccines we are getting in. And you can see the view from the top. Now, let us do a little more a thinking kind of process. Okay. Now, this is the variable region, and this is the constant region. This region doesn't change. The top one changes. And that's the one virus is using to make mutations that will enable them to come into newer forms. What would be a good strategy here? Because one side we are making medicines for the variable region, other side has a constant region, and the variable region we have to make medicines every year. Now let's look at it more carefully here. It's a variable region, vaccines every year. What will you do? Anybody out there? Which region you can use it to make it a better vaccine? Yeah. Use the constant region. Yeah, that is right. If you use the constant region, what will happen? See, this is a constant region. Therefore, people are making, this is a current research going on. Okay. They are making this. And what's the advantage? See, I'm looking at it from the different view. This is from the top view. Therefore, we are making the medicine for the constant region. Now, look at this. This is the variable region on the top, constant region on the bottom, and the antibodies are attached to the constant region. Now, let's look at the virus. What are the benefits? There's a virus here. What will the virus do? They will use the constant region to make a new antigen. They try to make another one. Then try to make another one. Make another one. Will we care about it? Because our antibodies depending on the constant region. Therefore, we will be like fighting with these guys. We don't have to worry. And if things work well, we may have to use only one antibody for several years. Or who knows, maybe for the whole life, if things work. Okay. Now this comes to the almost the end part of my talk. And I want to summarize what we learned. We talked about immunity, the fancy immune cell, and we saw different players in the immunity field. Remember T cell, mast cells, things like that, basophil. And we talked about the inside part of the radio, and we had a quick look on the phagocytosis, and we saw a video of our cells killing the methicillin-resistant bacteria. And we talked about the thymus elementary school, where all our tissues are learning different aspects of our body to enable them to identify differentiate between our body and a foreign body. And there is something like, see, I know, sometimes some of things I learn, I forget fast. Some of things learn better. Some of our students learn for a long time. Same thing happens here. Some of these guys can learn longer. For example, measles vaccine, you take only once in a life, but flu, you take in every year. Therefore, some of the things can learn better or memorize better. Then we talked about the antigen presentation to the YAMA, and then we said it's important, learning these things important for cancer and different disease. Then we talked about antibody in depth, because antibody is the most important thing. Then we talked about this, the flu season. We said if we use the constant region, we can get a better medicine. It's a research currently going on. Okay. Now I want to talk about few things related to GYRA. And GYRA is a US-based organization, and there's another organization called STEM for Cells. We work together. This is a US-based organization for STEM area, and there is a challenge right now going on. It's called STEM for Creativity Challenge. And if you are students in any area, you don't need not be GYRA students. Any kind of students can join here. And we have the International Research Conference for Children 2024 happening in July 12 to 14. It's right time for registration. Please try to register, and you can present your work there. And we also have Transition Institute for Special Education, Special Needs Children. Thank you all for your listening, and I'll be happy to take any questions. Should I stop sharing or?
Yes, you can stop sharing, Dr. Mathews. So there will be a 20-second delay to get questions. But I have a question. So
Yeah.
you mentioned about 5 different types of antibodies.
Yeah.
Among those 5 types, which one do you think is the best for developing vaccines or therapeutics, and why?
Oh, there's if I would say, it will be the ones involved in the most of the attack, that are alpha ones. because now they're in the phagocytosis, things like that. But if you develop things, then probably if you learn the process in that, probably it can apply for more diseases and things like that.
Okay.
Yeah,
And
thank you for
then
the question. It's a good question.
Okay, thank you. And there is a question in the chat in the Zoom. Can you explain how you crystallize or how you identify the antibodies? What is the method which you use?
Yeah, if you are looking for the studying the molecular level of antibodies, we have to use X-rays. See, X-rays are something you can see even atoms there. Okay, X-rays have very, very short wavelength, which enable you to see very, very small things. For that purpose, you have to crystallize that antibody. Then once you crystallize it, you have the system crystallized in one certain position. And when you pass the X-rays, you get the signal out. From the analysis of those things, you can get individual positions of these atoms. Once you have the position, then you can create the whole model. From there, you can study by using various things, like you can interact that antibody with the antigen or things like that and see how they function. Once you know how they are functioning, then you can develop things to make it function better or to clamp it down to stop it. Thank you. Hope it is clear.
Yeah, I think so. That means this field is purely interdisciplinary. That means biology is composing of
Yeah.
physics, chemistry, mathematics, and nowadays computer for modeling.
Yeah, exactly. And there are our studies also we do in a different ways. Some techniques you can use for specific this one, like Pinot's technique you can use in a different system, like in a bigger system. And there are solution studies where you can see the dynamics of these proteins. You have to combine different things to really get a grasp of what is going on.
Okay, thank you.
Thank you.
There is a question from YouTube. Why multiple doses like boosters are required for COVID vaccine?
Yeah, that's a very interesting question. One of the reason is with COVID, there are various changes happening. See, the way I was talking about the variable region, COVID also has that kind of regions, and they can mutate. Mutation is the main culprit for need, and they're expecting these things. And when you have a newer vaccine, they're going to take care of the newer modified viruses, because virus knows what's going on in the system. Therefore, next time when the virus mutates, they make sure they can attack you better. Therefore, you have to be prepared. Now, if you think about the ideas we're talking about antigen antibody reaction, if you know very clearly how this reaction is happening, even the virus mutates one thing, you can immediately make a compound that can block that mutation. You see? Therefore, if that's why they try to put more and more booster shots. But no, this is the COVID. The problem is things were happening so fast. We didn't have enough time to really learn everything. And people are still trying to make better medications. Thank
there
you.
There is another question in connection with COVID that is also from YouTube.
Uh-huh.
What is the antigen test used for COVID-19 detection?
Yeah, here because once you have the system coming in, it makes some of the things from the body reacts. From the antigen, body reacts with the response for the antigen. From that, you can find out whether this antigen is there or not. That's what they're testing. They're testing for the antigen presence in the system.
Yes.
Thank you.
Is there any unit to measure immunity? What is the scale or what is the metric to assess the immunity? That's also a question from YouTube.
I don't know exactly for that answer, but I would say there are different, if you go to the doctor, there are different indications to see whether you are healthy or not. And for example, you may be getting headache, that may be due to some reaction, or you may be having fever. Most of the fever is good, but sometimes that fever is due to an infection from the bacteria or things like that. Therefore, I don't know. To me, it's a little bit tricky to exactly say what immunity is. As long as you are healthy, that means you are immune to most of the things. Thank you.
Yes, yeah. Probably the question might
You can add,
be,
yeah.
Question might be related to how to measure the antibodies, probably the titer, and
I see. Yeah, I see. Yeah.
that could be the one.
Mhm.
Okay. If we are able to see as small as atoms,
Mhm.
why still patients are asked for ultrasound and MRI? That's a wonderful question. That's also from.
That's okay.
Yeah.
That's very nice. So for seeing the atom, you have to isolate the protein and do it in your crystallize all those things. Therefore, in the MRI system, the body, you can't see the atoms in the body. Body is a bigger system. Therefore, you have to use techniques which you can see the information. Like when you go to the airport, they do the X-ray scan. They can't to see these things. They can't cut out and then open and do things. This is similarly the idea. If in the body, if you have to take the protein out, then you will be in a really bad shape. Therefore, they use technique which you can keep the system stable and at the same time find out what is going on. Therefore, they can't really go to the atom level there. Thank
And
you.
also
That's a very thought-provoking question. Very nice.
yes, and also probably the time constraints for identifying the atoms and all those minute particles through XRD and other techniques, it may
Yeah,
not be able
that's true.
to translate to a clinical scenario. Yes, thank you.
Exactly.
Dr. Mathews, another question is, a person who is able to remember everything for a long time, unfortunately has a problem with remembering the names of the people and places. What is the reason behind this? I don't think this is onto your area, but still,
Yeah.
if you give some insights, that will be nice.
Yeah, as you finished it now, I probably don't know much about it, but I know the brain has different areas for different things. So depending on the which area is more used, that may be training better, or depending on which area is weaker, that part may be like for me, like sometimes I don't remember the names very well, but I remember some of the mathematical or scientific concepts. For there varies between the area of the brain maybe, but I am not the right person to talk about that. Thank you.
And I think we have time for a couple of more questions. Are there any molecular docking models or systems available to predict antigen antibody reaction?
Yeah, there are various things available. And computation, for example, when we were doing the COVID research, we had a really big computational group using supercomputers to predict how the models are happening. Because to do the X-ray kind of work, you have to isolate the protein. That needs a lot of time. If you can model it using computers, now because the computing power, you can even model it at the molecular level. And some more things comes really good. Therefore, that's a very important area, and modeling helps a lot for the
Yes, sir.
identifying.
Do you have any model in mind, any programs in your mind to help that question?
Yeah, there are modeling. Some of the softwares are very specific because because some of them you really need to pay for. Schrodinger, that group has a package of um, programs which can use for different things. And ICM, there are different programs, but most of them you have to pay to get it. That's why. But big academic institutions will have these things.
Yes. Yeah, and before we go for the last question, there is a question in the chat. The chair was mentioning about your discovery related to cancer. Can you just elaborate a little bit on that?
Yeah, here what we did was there is a receptor, AXL receptor, that is involved in making bodies who do cells do migration, and they can make it a survival. All those things are activated by this receptor. And this is exactly what when cancer cells are needed. They want to migrate, they want to survive, they want to move around, all those things. And what we found was if we block that receptor, then metastasis can be stopped. And blocking is not really easy because these are protein molecules which they don't have any, these proteins, these are binders. They don't have exactly any active site. If we have to make a model which looks like a protein, therefore we made that. And the other issue we had was these protein molecules bind really well. And we have to get a better binder. By then doing a lot of mutation studies and various things, we were able to get a really good model which binds 1,000 times faster than the actual ligand for the axle. This when the axle starts, when the ligand binds to it, then the migration and everything starts. If we block this using a better ligand, we made a ligand, and that one showed really good efficacy in the real animal models. That's what we got that FDA approval for that, because of the importance of that, to stop metastasis. We were basically concentrating on ovarian cancer, and it's very successful kind of way. Thank you.
Yeah, the basic idea is to fool the immune system or the biological system with a chemical molecule which you synthesized in your lab.
Yeah, it's our collaborators. Yeah.
Yes, cool. Yeah, thank you.
Yeah.
And yeah, before we wind up, can you give a little bit about Stanford University and how a aspiring scientist like the school kids or whoever is in the college level can get into the world's top universities such as Stanford, MIT, or University of California, like that. With that, we will wind up this Q&A session.
Yeah. Stanford usually looks for people with high talent. Like, for example, if a student is coming from the GYRA background, because if you have done research already, then those kind of people may be the ones Stanford is looking for, because they're looking for special talents. And here I see a lot of people from different parts. Some of them are from IIT, they are gold medals, things like that, and they come in. And from coming from our place, even at a higher school level, college level, you can get admission here when you do something very interesting. And they look for community kind of work and scientific area, or any kind of even literature, various things they can support. And Stanford has really good background. Before the Stanford site, it was a barn, and people used to mock at them because they converted the barn to a big place where all sorts of scientists are living. And actually, they have around 21 Nobel laureates currently living at Stanford. And it's a really good place, a lot of academic activities. That's what I see mostly happening here. And if you're interested, you will get it. That's what I think. If you aim for something, you will reach there. And again, I said, no, as long as you have the passion to drive that aim. Thank you. Hopefully, we'll see you here.
Yeah. That is so amazing that 21 Nobel laureates are currently working in Stanford University, and Stanford University has created, I think, 87 Nobel laureates in
something like that. Yeah.
Last few years. Yes. Thank you, Dr. Irimpan Mathews, for your wonderful talk, for your insights, and also your encouraging thoughts and words that I'm sure have encouraged hundreds of students in our Zoom platform, as well as the people, all students who are watching in YouTube. Thank you again. Wishing you all the success, and wishing you all the success for your future. Thank you.
Thank you for letting me speak here. It was nice interacting with all. Yeah. Thank you.
You are muted. Vidyadhiraja school is muted. Can you unmute and speak?
Thank you, sir. Let me invite Mr. K.N.S. Nair, President Vidyadhiraja Education Society, to felicitate Dr. Irimpan Mathews with a virtual memento.
Dr. Irimpan Mathews. Speak. Thank you very much for your wonderful talk. You have made it so simple for even a layman to understand. Thank you very much. This will be very much useful to our students and the entire audience. And we look forward to much more talks from your side. Thank you so much from the Vidyadhiraja family. Thanks a lot.
Thank you.
I think we can move on to the second part of the symposium, that is the presentation by the students. We have 3 teams ready to present. They are all GYRA scholars. Whoever is watching us, please be aware that this is not a complete project. They initiated this project 3-4 months before, and this is still in the conceptual level and hypothetical level. But still, they are very enlightened to share whatever they have so far with the vibrant audience. With that, there will be 3 presentations. They will be talking about 7 minutes, and we will be having additional 3 minutes for some quick question and answers. You can see the email IDs of their mentors, their teachers, in the last slide of their presentation. If you still have any questions, feel free to reach out to them via email. They will be more than happy to answer your queries. With that, let's move on to our first presentation. Back to VHS team.
Let me invite Master Samadhan, Master Sahil, Ms. Sejal Rana, and Ms. Sejal Gupta with the project on environment and earth sciences.
Good morning, everyone. Today we are here to present the project on the topic of environment and earth science, upcycling the floral waste accumulated in Bhandupeshwar Lake to reduce its harmful effects on aquatic life and its immediate environment. Our team members are Vaishnavi Gurak, Sahil Dole, Samadhan Bharatwat, Sejal Rana, Sejal Gupta, and Gayatri Bavaswar. Our advisor is Dr. Fino, and coach is Ms. Helen Roach. As you can see, there is a beautiful picture of Bhandupeshwar Lake in the background of this slide. This is the introduction of our project. Bhandupeshwar Lake is popularly known for its beauty and livelihood. It is the best destination for celebrating festivals. Their livelihood is based on fishing. Bhandupeshwar Lake is located near Tata Nagar, Bhandupesh, near Vidyadhiraja High School and Junior College. The area of Bhandupeshwar Lake is 0.1 square kilometer. Population of Tata Nagar is 840. Sejal, please continue the session.
These are some findings about the Bhandupeshwar Lake. As we can see, a man is throwing floral waste in the pond, and this is the floral waste being disposed in the Bhandupeshwar Lake. Let's see how floral waste affects the ecosystem. Floral waste disposal creates problems like eel and worm development. Reports have demonstrated that the death of the fishes in the Lake Naivasha at the beginning of 2010 was not because of chemical substances, but from the disposed flowers. A study found that over 50% of respondents who worked in flower farm were reported at least one of the symptoms of pesticide exposure, such as dizziness, nausea, diarrhea, and headache. Flowers are rich in nutrient content. The process of digging leaks nutrient leads to algae growth. Algae blooms in the river decreases in oxygen content. Reduced oxygen level can be harmful for fishes, leading to stress or suffocation. Flowers are not decomposed properly because of the mixture of some non-biodegradable waste. Flowers release nutrients during decomposition, affect fishes' health. These are some fishes found in the Bhandupeshwar Lake. The ordinary names of the fishes are at the top and the scientific names are at the bottom. Tilapia, Oreochromis niloticus, Late Scalcarifer, Barramundi, crystal-eyed catfish, Hemibagris vaiki, crab, Brachyura, catfish, Siluriformis, prawns, Dendrobanchiata. Over to you, Sejal.
Thank you, Sejal. We read several articles and we found this data from the previous study. Disposal of flowers in river affect the living organisms present in the water. Floral waste not only clog up with water channels, it also releases pesticide and insecticide into the already murky water. Flowers thrown in pond by human will be very harmful for human and animal health. These are some upcycling products that can be made from floral waste, including oil extraction, perfume, incense stick, and soap. Samadhan, please continue the session.
Thank you, Sejal. Based on the observation, we have formulated problem statement for our project. Floral waste in Vandupeshwar Lake causes harmful effect in aquatic life and its immediate surrounding. Based on the observation, we have formulated hypothesis for our project. Understanding the harmful effect of floral waste in water resources and creating awareness of floral waste management pave way for innovative solutions for upcycling floral waste at domestic level. Based on the hypothesis, we have formulated objectives for our project to identify the harmful effect of floral waste in aquatic ecosystem, to prepare socially useful product by using household floral waste, to create awareness among community regarding the floral waste management. Sahil, please continue.
Thank you, Samadhan. This is the methodology of our project, Floral Waste Management. We are going to conduct a survey. The answer will be received from people through survey. Based on that, we are going to conduct an awareness program. We are going to educate people regarding healthy floral waste management practices. After that, we are going to do an experiment regarding impacts of floral waste. To prove environmental impact, we are going to collect water from the pond and pH level will be checked. After that, we will put some floral waste in it and keep it for 3 days. We will observe and check the pH value of water each day. After that, to find out how fishes are affected by floral waste, we will put Pseudodeformas and tilapia fishes in that water and observe the lifespan of fishes in that polluted water. Next, to check microbial contamination, we are going to make use of check and CK. To check pH value, we are going to make use of litmus paper. To check hardness of water, we are going to make use of TDS meter. In order to upcycle household floral waste, we will make perfume, incense sticks, and extraction of oil. We expect the following results from our project. A well-informed community regarding healthy floral waste management practices will help us to grab attention of people towards healthy floral waste management. Evidences regarding environmental, microbial, and quality impacts of floral waste will assist us to concentrate more on harmful effects of floral waste in water bodies. Sustainable upcycling method to manage floral waste at household level will assist us to grab attention of people on the topic how to convert household floral waste into socially useful product that can even help them to build a small ordinary business at home. This is ongoing study of our project. We are preparing questionnaire for survey. We are reading articles regarding upcycling floral waste. Moreover, to make following products, incense sticks, oil extraction, and perfume. We are finding out a new process to innovate these products. Now I would like to express my heartfelt gratitude to Team GYRA for guiding us and Mrs. Sangeeta Bankar for helping us to get information about Vandupeshwar Lake. Let me conclude this presentation with a wonderful quote of Leonardo da Vinci. Water is the driving force of all nature. Thank you.
Thank you, friends. It's time for the question answer session.
Thank you for this wonderful presentation. I would request our eminent speaker to begin with question if you have any.
Yeah, thank you. Thank you, Pinocho. Thank you for this beautiful presentation. It's really good, very clear and very nicely coordinated. I was thinking, when these people celebrate there's a lot of chance for more and more flowers coming in. Therefore, your teaching process probably need to have something related to the people's celebration, like how to we can't eliminate bringing flowers, but maybe there are different strategies for that one. Then regarding your experiments, you're using a TLD meter. That's very nice. And now we talk about, you said the oxygen content is going down. Are there any plans to check that in your test procedures or see whether oxygen content, measuring the oxygen content in the system? Are you planning that one, or is it going to be useful? Or you are muted.
No, you are not muted. Come closer to the mic. Come close to the mic. Yes, you can speak now.
Thank you, sir, for the question. We have not planned for that.
Okay, thank you.
Yeah, there is a question in the Zoom chat. Sorry, let me scroll it down. Okay, why did you choose this Bandupeshwar Lake for your studies?
As it is near to us, and we saw problems in it regarding floral waste management, and it's the representation of Bhandupis, so we chose this Bandupeshwar Lake.
Okay, that means the studies in this lake which you are conducting can be replicated to other places as well, right?
Yes, sir.
Okay. There is another question again. Why the flowers are considered to be toxic? Are they? It seems like they are good, right? But why?
Thank you for the question. In the flower farms, farmers are putting pesticides to grow the plants. So that will be toxic to aquatic animals in the pond.
Okay, that means when people are dumping more flowers over there, pesticide level in that area is increasing. That is what your concern is. I hope the question is answered. I don't see any questions in the chat after that from YouTube. It seems like we can move to the next session. This is very wonderful work. You are trying to identify the real root cause of a simple problem and trying to find a remedy for that. All the best. Thank you. And Dr. Mathews.
And one comment, because some fishes are more sensitive for the toxins. And Finosh probably knows that. Therefore, when you study the fish, maybe you have to select the right fish in that family. Yeah. Thank you.
Yeah. Before you leave, I have one question. Is there fishing in that lake? People eating those fishes?
Yes. Thank you, sir, for this question. Yeah.
Oh, yeah. Yes. Okay. Good job. Thank you. All the best for your future studies.
Thank you,
We
sir.
can move on to the second presentation.
Here we have 5 members to present a project on educational impacts that affect the personal and overall development of the underprivileged communities in Somte, Palghar district.
Good morning, honorable present here. I am Venkatesh from Team B. Our title is Educational Impact that affect the personal and overall development of the underprivileged communities in the Somte village, Palghar district. Our area is social science. Our coach is Mr. Sharad Upalkar. Our team members are Bhargavi, Komal, Mathews, Samyak, Shaurya, Venkatesh. Now I invite Bhargavi to start the presentation.
Thanks, Venkatesh. Now let us begin with the importance of education. Education is important for a society. It creates more employment opportunity. It helps people to secure a higher income. Education improves the overall economy of a region. It is important to lead a prosperous and happy life. People can give back to their community if they are educated. It also makes one independent. Urban areas in Maharashtra have good educational facilities for students. However, rural areas are undeveloped in the terms of education. Indian government has made several policies for tribe education in villages such as Eklava Model Residential School, introduced in 1997 to provide quality education to scheduled tribe students, and National Fellowship and Scholarship, provide financial assistance to scheduled tribe students for MPhil and PhD. Still, underprivileged communities do not receive proper education facilities. They don't have proper schools and libraries. There is shortage of teachers, and people don't have much awareness of education and its importance. One such village is Somte village, located in Palghar district of Maharashtra. The total population of the village is 1,761, out of which 777 are literate and 987 are illiterate. The total number of children is 326 The geographical area is 655.93 hectares. The literacy rate of the Somte village is 44.12%, out of which 53.30% males and 34.64% females are literate. Here is the brief background of Somte village. There is only one school in the village, ZP San Somte School. The school has no facilities of education, and the people don't have much awareness of education and its importance. Our research is to study the quality of education in Somte village. The purpose of our study is to understanding the educational need in Somte village, Palghar district. Ved, please continue.
Thanks, Bharat. Palghar is a progressive district of Maharashtra. It has a mixture of tribal and rural population. The livelihood of the people is based on the forest and agriculture. The people have a strong attachment to food. The major sources of employment are farming, collecting minor forest produce, dairy, making bricks, fishing, etc. Agriculture is the main occupation of Palghar district. The people contribute to the economy through ecotourism. According to the study, school dropout and inquiry in the rural areas, Palghar Taluka, 88% of students stop going to school due to poverty. Children engage in domestic work. They are interested in studies, but sadly, they don't have facilities. Parents hesitate to send their children to school. Jilla Parishad school teaches in vernacular medium. Madhu, please continue.
Thanks, Ved. Based on the data collected, we have come up with a problem statement. Underprivileged communities in Somte village, Palghar district, lack quality education which affects their personal and overall development. This is the hypothesis of our project. Understanding the reasons for poor quality of education in the underprivileged community and its impact on their life help us in suggesting improvement and creating awareness among people regarding the need of education. These are the aims of our project. Our aim is to identify the factors depriving the children of education in Somte village. We want to find feasible ways of supporting education with the help of local organization and to create awareness among the villagers regarding the need of education. We will make recommendations to the authorities in improving educational needs in Somte village. Shaurya, please continue.
Thanks, Madhu. Here is the detailed methodology of our project. Our first step will be to conduct the surveys, serpents, teachers, parents, and children. After conducting the survey, our team will analyze the data collected. Based on this analysis, we will be coming up with recommendations to the authorities for improvement in the existing education system. Our final step is to create awareness among parents and children about the need to get educated. These are the questionnaires prepared by the team to interview the serpents, teachers, school administration, parents, and children. These interviews will help us in gathering details about the existing educational background. At present, our team is reading an article on Palghar district to know more about the underprivileged communities. We are also reading government policies introduced to ensure tribal education. And we are also revising the questionnaire prepared for the serpents, teachers, school administration, parents, and children. Our study gives evidence regarding the poor quality of education in Somte village. It helps the team to give creative recommendations to reduce the dropout. The study inspires the community towards the path of education. Acknowledgement to Team GYRA for guiding and encouraging us, Mr. Sanjeev Kumar for helping us to gather information about Somte village. Let me conclude our presentation with a meaningful thought by Alan Lee. Education is the movement from darkness to light. Thank you.
Thank you, friends. Inviting the audience to come up with questions.
Thank you. Thank you, team, for your wonderful presentation. I would begin with Dr. Mathews.
Yeah, so really beautiful. And I don't know, but question-wise, you really covered it very well. One of my thinking is, when you are a student and other school has problems in education, maybe you yourself can get involved in teaching. Like, you can take school different groups and try to go and teach them at the basic level. By the time you get the results from your studies, you may be getting some students ready there to work hard on these projects. And thank you for involving the parents. I think that is the best way to get the authorities look into details. Thank you. It's really nice Nice work. Thank you.
Yeah, I have one question related to your study. Why did you choose Somte village? The same question, it's a replication of the same question which the previous team have answered. So you have any specific reason to choose Somte? Yeah, come forward and
Because Somte village is one of the nearest village to Mumbai, and one of our team member has got to know about the village, about the village situation. So that's why our team started to study about it for our village.
thank you. There is a question in the Zoom chat. So if your study is a successful one, how this is going to help those people in Somte village? That means I can simplify the question. So what you are going to do for students at Somte village? You mentioned there is poverty, there is lack of education, there is lack of awareness. So what is your action step? I think that's what the question is about.
Thank you for the question. It helps to increase the standard of living of the village.
That's what you are going to achieve.
Yes.
Okay. And then how are you going to conduct the questionnaire? You made questionnaire, right? So how you are going to collect the data?
By visiting the village.
You will be visiting the village?
Yes, sir.
Oh. Okay, thank you. I don't see any question from the YouTube.
One
Yeah.
other question. See, suppose you went through all this process and got the village really good. Are you going to stop? Are you going to plan for other things? Or suppose your project is really successful and you make the village really good, like an education system, and they don't need any help anymore. What will you do next?
So the question is, if you are successful in this project, you identify what have happened in the Somte village which limits their education. So what remedies you are going to suggest or recommend, or you as a student group going to help those kids with your findings?
Yes, sir. We will think about it.
Okay, cool. Yeah, if you think in futuristic aspect, because this is not a simple study and you are not finishing the study, but instead you need to do something for their welfare. So you as a team of group of students, you can do many things. And there is one question, how did you come to this problem? I think this is from you too. How did you come to this problem? Why you choose this problem? That is, okay, you can come forward because the mic is not capturing. If you are speaking, come forward. Okay, no problem. These are the, if you have any answer, you can mention. If not, no worries.
My father shared the details about the village.
Okay, so somebody told that there is a village like this and the people here are not well in
Yes.
getting education. And okay, cool. So these are the fundamental questions. See, when you present to a broad audience, you are getting so many questions. This is not the end. This is the beginning. Yeah, don't worry. Think about this. Try to identify answers. Try to find answers for these. You are a very good group, wonderful group. So discuss among yourself and come up with solutions for these. Okay, once again, all the best. very nice work. You can do many things for Somte village. And thank you very much. We can move to the next presentation.
Thank you,
Thank you, sir.
The team members are coming up with their project based on art and culture.
Good morning, one and all present here. I am Tanuja from Team C. We are working on stress management among senior secondary students. My team members are Liana, Kavya, Tanuja, Vaishnavi, and Anu. Our coach is Mrs. Babita Anand. Our advisor is Dr. Vinod sir. The title of our project is Artistic Activities Reduce Stress among the senior high school students of Vidyadhiraja. I will now explain what stress is and how it can impact our overall well-being. Stress can be defined as our physical and mental responses to particular events. For example, getting ready to go out for a party or something bad, such as an argument with your family members or feeling tense for a test. Stress can also affect our academic life. Trying to please teachers or parents with good grades can also be a reason for stress. It can feel overwhelming. Social anxiety is the third most common mental health condition. It is one of the impacts on our mental health. When we have social anxiety, we can feel judged by people. At school, it can be hard to deal with friends, bullies, etc. Friends are family, right? Kavya, can you please explain how family conflict can affect an individual?
Sure, a family conflict can affect a teenager's life immensely. Family issues such as parents divorcing, financial issues, or not getting along with siblings will also contribute to stress. Have you ever felt conscious when you joined a new school? It's a big change, right? Big life changes such as moving to a new town and changing schools can have impact on your mental health. This is some information we collected from the Organization for Economic Cooperation and Development. OECD conducted a survey across 72 countries. 540,000 students attempted the survey. Here is a pie chart telling us how much percentage of students are stressed. The causes are also mentioned. These are the reasons why most of the students are stressed. You may or may not relate to these reasons, but it's okay if you do. I will now hand over the presentation to Vaishnavi.
Thanks, Kavya. Here is our selected problem statement. Academic pressure is increasing among the senior secondary students, and the inclusion of art activities would reduce the stress. Based on our observation, we have come up with the hypothesis of our project, analyzing the reasons for stress among the senior secondary students and incorporate art activities in their curriculum to minimize after effects of stress. These are the objectives. Identify various factors that create stress among the senior secondary students, to provide the senior students with art activities, to make recommendation in incorporating art integrated curriculum for the senior students. Diana, please continue.
Thanks, Vaishnavi. Here's our methodology. Our first step is to conduct a survey for the high school students. Then we will be making an analysis. We will note down and conduct the activities preferred by students. A post-activity survey will be conducted to check whether their stress has reduced or not. Here is a survey that we will be conducting for the high school students based on the following topics: academics, art, anxiety, stress, overthinking, coping issues. Right now, we have been reading some scholarly articles about stress. Our team is also studying the stress surveys conducted by UNICEF and OECD. In addition to this, we will be revising the stress questionnaire prepared by our team. These are the expected outcomes of our project. The use of art as a stress relief for the senior secondary students will be beneficial. A youth aware of the negative effects of stress on life. Upskilling the youth by providing them with new and effective methods of coping with stress. Recommending the school management to include stress-busting activities for the senior secondary students. We acknowledge GYRA for guiding and mentoring us throughout.
Now, I conclude our presentation with this wonderful thought by Leonardo da Vinci. Learning never exhausts the mind. Thank you.
Thank you, friends. Our team members are ready to answer your queries.
Thank you, team, for this wonderful concept. And basically, this is the immediate need of the hour, because students in everywhere, whether it is in US or Europe or India or anywhere, students are always found to be stressed, especially when they are anticipating some examination. Great job, great concept, great thought. Dr. Mathews, you can start with your question.
Yeah. Thank you. Very nice job. And you are attacking some really important problem. And for stress analysis, you're trying to check with the students about the stress and trying to have some kind of art activity. How do you rate the activity with the students? Because you are going to use one classroom for the approach, or you have a group of students try to use the art activity, and how do you try to, because some students may like some art activity, that's what you were mentioning. Are you going to group those kind of students together and study, or you have a diverse student population and study the art activity? What will be your preference?
Did you understand the question?
Yes. The students are of separate standard, so we'll conduct the activities preferred by them in their respective classrooms.
Okay, thank you.
Okay, there is a question from the YouTube. How can you make sure that they get proper facility?
Sorry?
For example, how can you make sure that they get proper facility? What I understood is, if you are proposing that if they can be given, or if they can help with some art related activities to relieve the stress, whether your school authority is willing to do that. How can you ensure, okay, your findings are benefiting the classes 8 and 9, which you mentioned? You got it?
Yes.
Yeah, you have any comments for that?
We'll think about it.
Yeah, you can recommend. You can recommend to your school management. Nair sir is here. He is open for everything. Just mention, hey sir, our 9th and 8th and 9th grade students are stressed when they come closer to their exams. Please do these things. These are our findings and these are our recommendation. You can talk to your principal, ma'am. So they will be helping to implement those things. Okay. And then another question, where are you going to conduct the survey?
We are going to conduct the survey on Google Forms.
Yeah, in Google Forms, right? So you will be distributing the Google Forms together. Okay.
Yes.
There is another question from the Zoom. It is actually two people asked more or less the same question. I can compile both. So why did you choose this particular class? Why others? Other people are not stressed?
No, actually, the extracurricular activities are only till 8th standard. After 9th and 10th, the academic pressure increases day by day.
Okay. Yeah, so that makes sense. Like till 8th grade, your school will be having activities for the students, extracurricular activities, but 9th and 10th grade, when they reach, they will be more focusing towards studies. That's why.
Yes.
Okay, that makes sense. Thank you.
Thank you.
Okay, one more final question. How are you sure that art and culture reduces academic stress?
We read an article by Harvard that today's generation doesn't really open up to counselors or therapists or any cure to stress and anxiety. So Harvard stated that art can be beneficial to reduce stress.
That is great. So I hope your answer satisfied the questionnaire. Thank you again. Okay, I think no more question. Okay, so this is again wonderful question. The idea, the concept, the problem which you chose, that is really the need of our. You can do many things. Again, the same concept, the same findings can be replicable to many other schools across the country, so that with this simple thought, with this simple intervention, you should be able to revolutionize the current system, and help millions of students to come out of their academic pressure and stress. Thank you again. All the best, all the very best. We will see.
Thank you.
Yeah, we are expecting many outstanding findings from you in the future related to this topic. Thank you.
Thank you so much.
We appreciate the support and participation of everyone involved in making this symposium a success. And now, Team GYRA will share their further announcements.
Thank you, Vaishnavi, for this opportunity. I have a couple of announcements. First of all, before going into announcement session from Team GYRA and on behalf of...
We appreciate the support and participation of everyone.
So please mute whoever in the Zoom. Yeah, we sincerely apologize for that technical glitch that has happened initially. So we were struggling to resolve it. And I admire the willpower of the teachers and students to come back with 100% positivity. Yeah, that's the takeaway message which I would like to share today. Yeah, I don't want to take much of your time. So I have a couple of announcements. One is we will be having another virtual symposium on 6th of April, same time. That's a Saturday from 11 a.m. to 1 p.m. The speaker is Dr. Vipin Dev. He is from MIT Harvard. The topic will be on geo-satellite communications and sensing network. I think that is going to be another different avenue, which is an emerging field in the science. Please make use of this opportunity. We will be sharing all the information through our website, as well as we will be sharing all those information through our YouTube channel as well. Please take a moment to subscribe our channel if you didn't do so. And also we have IRCC, International Research Conference for Children, which is scheduled on July 12 to 14 at Assisi Vidyaniketan Public School, Ernakulam. So we are expecting 1,500 students from different parts of the world, and there will be plenary talks and keynote addresses by at least 15 to 20 eminent scientists from multiple areas of research, and there will be more than 100 students, teams will be presenting. So please do register, because we may have to close the registration when we reach up to 1,200 or 1,300, because we don't have enough facility to accommodate more than 1,500 students. So please don't delay for your registration. Keep your registration, and you can submit abstract if you want to present any of your research findings. So that's all from our side. And also, if you want to join GYRA, please email us. Please visit our website. Our coordinator will be reaching out to you to take forward for the next step. Thank you. And back to Team VHS.
In the words of Thomas Edison, genius is 1% inspiration and 99% perspiration. We acknowledge the brilliant insight shared by our keynote speaker, Dr. Irimpan Mathews, and the hard work showcased by our young researchers. Let's conclude this event with a vote of thanks. Vaishnavi, please. Distinguished guests, participants, and viewers. As we have come to the conclusion of the virtual symposium, it is my privilege to express our heartfelt gratitude to all who have made this event possible. Our sincere thanks go to GYRA for providing VHS with the opportunity to host the symposium. Yes, the support has made it possible to create a platform for learning, curiosity, and exchange of ideas. Special appreciation to Dr. Finosh. Your role as the chair of today's event has been invaluable, guiding us through the symposium with poise and expertise. We extend our deepest gratitude to our distinguished guest speaker, Dr. Irimpan Mathews, sir. Your insights on extreme view of immunity have broadened our interest in science and research. We are immensely grateful for the knowledge you have imparted to us. We acknowledge the guidance of Nair sir, whose leadership and commitment to educational excellence have been pivotal in shaping our school community. To our coaches, audience, students from various schools and teachers, we extend our heartfelt appreciation. Your active participation and engagement have been the driving force behind the success of the symposium. In closing, we extend our sincere thanks to everyone involved for their contribution in making this event a resounding success. We will see you all in the next symposium. Thank you.
Thank you again, and the meeting is now concluded. YouTube streaming can be stopped now. Thank you.